I want you to look at me and think, I'm going to try to explain. It's actually complex. Hopefully, I'm going to do a decent job in transferring what I want to transfer. I'm going to start by saying how we do solve problems, how we solve problems as scientists. But before that, I'm going to give you a very simple quiz to see how you think. Maybe we have potential scientists in the audience. The first thing is very simple. If you know that, don't destroy for the others, because I took it from the internet, so it's easy to take it, right? But the thing is, and this is true, just imagine that the car is fallen into the river. And you have a task, simple task, to take the car from the river out. A car has fallen into the river. You need to take the car out of the river. What are you going to do? Be creative. Crane. That's a good idea. Okay, I'll give you a hint. Actually, in a statistical test, people score, 95% of the people score the same idea, and it's simple. Bring a bigger car, right? So you have this car here. Oops. Yes, sorry. You have this, maybe this is, okay. You have a small car, and you want to take it out from the water. So you bring a bigger car. Very logical. However, only very sharp people, or physicists, maybe mathematicians also, can actually see now that the mass of the car, this is a physical question, is not as it seems, because this car is full of water. Therefore, the momentum is not appropriately calculated. And what will happen is this. Oops, sorry. It jumped, yeah. Okay? And now we have 2 cars in the water. Ideal solution? Bring a bigger car. Exactly. And this is the end of it. Right? So it's jumping a little bit here. I don't know why. This is the car. This is a bigger car, and that will happen. So history is coming back. It's a cycle, a vicious cycle, and we have to break the cycle. And this is the cycle, basically, of how scientists also think. So cancer is a complex disease, really complex, multidisciplinary. You need different approaches to try and solve it. And we are not naive. We're not going to solve it. We may be able to find better cure. We may be able to basically expand dramatically life expectancy. But cure is a bit of an issue here. And it's super complex, and I'll try to explain a little bit how we think. I want to start with my story. So I lost my mother when I was 18 years ago to cancer. That was more than 20 years ago. And then I decided that I will devote my life to fighting against cancer. 20 years later, I also lost my father to cancer. So you see, I don't have any other choice. I'm working for myself. And that's a great motivation, right? I have a bad genetics, apparently. Okay, so a little bit about our lab. We are totally in line the idea of the immune system. Our immune system is very important, and we are trying to understand and create new drugs based on how to manipulate the function of the immune system. That means that we are very active in everything related to immune system. That includes hematological malignancies. We are very active in multiple myeloma, in chronic lymphocytic leukemia, in mantle cell lymphoma, and in some others. And in inflammation, autoimmune diseases such as Crohn's disease and ulcerative colitis, which are also known as inflammatory bowel diseases, and also in some viral infections that affect the immune system, such as HIV. However, there are 2 more diseases that are called solid tumors, such as ovarian and brain tumor, glioblastoma multiforme, that also have an immune component. So, and also some rare genetic disease, and I don't have the time to actually go after all of them. In a nutshell, Our lab is divided into two major areas. People that are doing drug delivery system, basically creating nanoparticles, very, very tiny particles, from materials that we all have in the body, like sugars or lipids. And we can put drug inside and even give them a direction, basically home them into the appropriate cell types. And we are quite good at this. The other side is to do some biology. And there are lots of genetic knowledge these days. I'll talk a little bit about this in the next few slides. The ideal situation is to find new targets from patient samples and then create new drugs based on nanotechnology and together, mix them together, basically tailoring the treatment. So before I'm going to jump into this, I have to create a kind of a common shared knowledge with all of you. And I'm going to start by a simple biology. So I found this in the most advanced website for biology, which is called Google. Yes, so I searched for a cell, and this is the image I found. Okay, so it's very nice to see. All of our bodies, all of our cells are basically the same. So this is the brain of the cell. It has a DNA inside it, packaged. This is amazing. You know how much information you have here? And for some reason, it will be transcribed to this single letter that we call the messenger RNA. And now people are developing drugs against this, and now I'm one of them, so I'm going to talk about this a little bit. It will be translated to a protein by this machinery that is called a ribosome. Adayonat, if you heard, is responsible for the structure, or one of the people who is responsible for the structure, an Israeli scientist who got a Nobel Prize in chemistry in 2009. And this is very simple, right? So just imagine that we can now play with different parts of this equation. We can play with the DNA a little bit, we can play a lot with the messenger, and we can even play with proteins. But if you're smart enough, you know that to get into the brain of the cell is very hard, because there are also membranes here, and it's a tricky job. But to get or to find a messenger that is somewhere floating in the cell is easier. So we now have the ability to sequence the genome, which is the DNA, the transcriptome, which is the RNA, and edit gene. Just to give you a sense, 20 years ago, when I was still a graduate student, a man came to our university, Craig Venter, and he said, I have a vision. I will sequence a human genome. It will cost $1 billion, and it will take me 4 years. 1994 he started, 1998 was the first copy of one single person who was completely sequenced. It cost nearly $1 billion. We can do it today in the lab, $1,500, 24 hours. So this is the technology. Technology is very fast. The question is, what can we learn from this? And since I have a problem, because, you know, I'm a scientist. So as a scientist, I want to tell you that I was intrigued from the fact that we can actually sequence the genome. And I decided to sequence myself. Stupid mistake. Because what I found is that one of my chromosomes, one of my genes, is inverted. So it's the opposite. It should be like this, and one of them is like this. So I'm a scientist. I look around, went into literature, talked to my friends. I have good friends. One of them is the president of Dana-Farber. One of them is the president of MD Anderson. It's good places. I even know a few people in Sloan. And in the Broad Institute, the president, I said, what's going on? They said, we never heard about this. First case. That's intimidating. And the reason is that it could be that I was, bad in math. in high school. That's maybe the reason. Fortunately, I can't tell it to my parents. It could mean that this is the reason that I was genetically imprinted to become a scientist. And it could be that a few years from now, I will have a devastating disease. Everything is possible, nobody knows. Immediately, I sequence my kids. Everyone is normal. I mean, they're really okay. They're like this. I'm the only one like this. I said, maybe my wife. Maybe she's no, she's also okay. So we have a problem. We have a lot of genetic information. We don't really know what to do with it most of the time. But the majority of the time, we understand issues, mutations within a disease. Breast cancer and ovarian cancer is easy to explain. Women that have breast cancer and ovarian cancer are often being tested if they have mutation in two genes, BRCA1 and BRCA2. And if they have a mutation, that increases the chances of the offspring sometime in life to get cancer. It doesn't mean that they will get, but it increases. Is it correct? I'm telling you, it's completely incorrect. The reason is that the screening is only for known mutations, not rare mutations. So we are learning day by day that there are other factors that can happen. So that brings me to another area which is important. I had this, okay, which is very important, which is related to this, and how we design drug based on this. But before that, you already know this slide, because you know that each one reacts differently to drugs. Let's assume one have a headache. One could get a Tylenol, and it will work. One will don't do anything, it will work. One will have a shot of whiskey and will be happy. So, I mean, we are reacting differently to drugs. We are reacting differently to drugs because we don't look alike. Look at yourselves here. You're totally different. Now, I'll tell you something I learned last week in New York, not far away. There are really 16,000 diseases. 16, 60000. But there are only 500 treatments approved in general. When I'm saying 16,000, this is 10,000 that everyone knows, and 50,000 rare genetic diseases. So our approach is to create drugs that the immune system is involved. We strongly believe, kind of an holistic approach, that everything is connected. The immune system can influence how we treat cancer or how cancer deceives the immune system. New drugs that are now appearing, that we call them immuno-oncology area, is basically try to shield back the tumors to the immune system because the immune system are now being evaded by tumors. So we strongly believe that there is all connection. Inflammation will induce cancer at some point, and the opposite. Cancer can induce inflammation. And we are trying to fit, kind of a personalized, but not really, fit the patient to the real drug. And how do we do this? So this is a schematic concept. It's not really easy. So basically, when you have a patient, and it's not important which type of patient, this is an example for cancer, but it's true for everything. Since we are here in the frame of cancer, that's a good example. We can sequence the patient and understand a little bit about the mutation. This is a lot of mathematician work. Learn about mutation in genes that are either overexpressed or down-regulated. Then we can design in synthesis new drugs that are based on the RNA. Remember this single strand in the cell. We can silence these genes, make them go asleep, or if we need those genes, we can upregulate them. We can deliver another messenger from outside. And then we need to package this because these are highly negatively charged molecules. They are big, big molecules, and we have to carry them. And what we are doing is designing in delivery systems, we are packaging them. So just imagine that you can now need to package something and deliver it. We are kind of a taxi drivers, right? We take, instead of people, we take drugs from place to place, tiny taxis, like submarines that are floating inside the body. And the idea is to find areas or cells that are really affected, and we can deliver those drugs effectively. But we deliver a new class of drugs. In the past, we thought that chemotherapy is the best. Now, we know that chemotherapy is really the best only if it's delivered directly to the tumor. And just to give you numbers, and I hate to do it because this is a propaganda basically, but when you inject to a cancer patient chemotherapy, the amount that is really reaching the tumor is less than 1% of what you inject. So what is the reason to inject it? Usually, the reason is the fear from metastasis, because those chemotherapy usually kills metastasis. So this is the only thing that we have. Yes, I'm running. Okay. Okay, so we are playing with this. I don't have a lot of time. I want to give you two short examples. Short. One is a brain tumor, glioma. It's a devastating tumor. Life expectancy is usually 12 months to 15. Nothing else to do except from taking out the tumor. Friend of mine, Zvi Cohen from Sheba Medical Center, is a neurosurgeon, asked me, let's do something together. We sequenced patient samples, and we found that they have markers that we can actually target. So there is something that is joint marker for everyone. And we package this in tiny, tiny particles, about 16 nanometer in diameter, using this technology, and injected into the brain. This is a tumor in a mouse. And basically show that we can prolong the survival dramatically. Ovarian cancer, a very bad example. You know, it's always mutating, it's always changing. So I would say that, you know, this is an example of 39 patients that have gene profiling differently. Here again, we try to package in a smart way the genes that can do it. Just to give you an example, okay, this is the technology, how we purify. This is an example without being scientists. Look at the red. Okay, the red is in the tumor. It's basically a new drug based on RNA. After you inject into sick mice that have ovarian cancer, and you can see that they go into the tumor area that penetrate. And if you look at the tumor volumes, you know, compared to the control, it looks astonishing. So we can block it. And this is the tumor that is formed. So it's not going down, but you can maintain it, and quite for a long time. What next? How we do clinical translation? How we take it to the next level? In order to take it to the next level, we need to scale up the production. We need to design special materials in a good manufacturing product. We have to select the patient. It will not be one fits everyone. It's a different approach. Safety test, dosing, and etc. And we have to work with companies, unfortunately. Fortune costs a lot of money to do this. But we are not expecting, as an example, Israel Cancer Research Fund to fund a new drug. That will cost billions, not one, more than one. We are grateful for the Israeli Cancer Research Fund and other people that help us and fund our research, not only me, but other scientists in Israel, to help us create proof of concept. If we have proof of concept, like the brain tumor example that I've showed you, like the ovarian example, we can go and convince pharma, big pharma, or biotech company that there is an interest here, and we should move to the next level. That's it. My message is nanotechnology is a great tool. RNA drugs may be the future, and it's all in the message, messenger RNA. At the end, I want to thank my group, people that are really doing a lot of work, very international from different places around the world, even Australia. the States, a guy from Sloan, and others, physicians, different field, engineers, biologists, chemistry, different types, and of course, the funding agencies. And here I'm going to talk specifically about the Israel Cancer Research Fund, and I want to thank again Susan and Landmark for their amazing contribution. And again, I'm here mostly because of them. So thank you all for listening. If you have any questions, I'll be happy to answer.